Hey y'all. So in this video, we're going to do a very quick overview of the fascinating world of insect brains and insect neuroscience. So you may have guessed, behind me is actually an insect brain. That's the brain of Drosophila melanogaster, also known as the fruit fly. Right? And the fruit fly is just this powerhouse in the world of neuroscience. It's widely used as a model organism, and there are a bunch of really great molecular genetic tools and for understanding how the brain generates behavior. So in this image, what the researchers have done is actually use some of these molecular genetic tools as well as immunohistochemistry to actually specifically target fluorescent probes to a specific region of the fly brain that's responsible for learning and memory, right? And so that's highlighted in green there. And before we dive into that too much of the detail, let's take a step back and frame all of this, why insect brains are important in the context of what y'all have been talking about. So over the last few sections, y'all have heard quite a bit about complex behaviors that are observed in the insect world. So we can take the waggle dance as a really good example. So you can imagine an individual foraging honeybee has found a great area for floral resources, and now it's returning to its colony, and now is performing the waggle dance using a series of changes in body orientation, vibrating its abdomen and wings to communicate where exactly, how far, and what orientation this floral resource may be. Right? So this is a really complex process run for the individual that found that resource is now turning it into behavior, and then for the observing bees that are actually taking these visual cues, maybe some olfactory cues, and will potentially go forage in that area. Right? And so this is all made possible through specific regions within the insect brain that allow for these behaviors. Another way of kind of stating this is, right, the insect brain integrates information about the environment and facilitates decision making, learning, memory formation, and the execution of behaviors. And the way that the insect brain takes in information from the environment is something that is familiar to us using sensory systems. Right? So they can take olfactory cues, visual cues, taste thing, gustatory, somatosensory, touch, and all of that information is taken from the environment, then passed to higher regions of the insect brain for processing, kind of classification of this information, assigning meaning to it, and that can lead to output behavior. Right? So here's a little example of that. So here, this is a video from the crawl lab. So this is a foraging bee. It's navigating through this really stimulating environment, lands on a flower, and then decides to leave. Right? So I think this is a really cool example of how insects have to navigate complex environments, filter out information that's not useful to them, focus on their objective. And again, it's through these sensory processing and information processing centers in the brain that allows them to do that. So this cartoon on the right kind of highlights some of the sensory organs that are important in this foraging event. So things like the eye filtering for specific shapes, colors, the antennae, right, which is processing olfactory information, guiding this bee to potentially look for a specific flower, right, and that information is again passed to deeper into the brain where it's coded, classified, and then eventually turned into an output behavior of, hey, this bee is going to land on a flower or not. All right, so next we're actually going to look at some data showing volumetric representations of these important information processing centers in the insect brain. But before then, we're going to do a very fast review of the most basic unit of the brain and nervous system, which is the neuron. Right? So the neuron, I think the most fundamental important feature of a neuron is being able to propagate or send information from one location to another. And the way it does this is in part using these dendrites, right? So they receive information either from other neurons, or if they're on the very most sensory periphery edge of your system, they could have different types of receptors encoded in them for different environmental stimuli, right? So think back to olfaction. They could have olfactory receptors encoded in the dendrites, right? And those in our insects would be located in the antennae, right? And so when an odorant binds to that receptor, it'll send information through that dendrite to the cell body, also known as the soma, right? And this integrates information from those dendrites, and once a threshold is reached, it will propagate that signal and with an action potential down the axon, right, which allows basically elongates the cell and allows a bunch of different connections through the axon terminal, which is where either you can have synapses onto other neurons, or your neuron will release neurotransmitters that can lead to functional change, physiological changes, signaling cascades, and eventually, right, this behavioral output. Right? And through these connections that we can form between neurons, called synapses, we're able to form more complex networks of neural tissue that can create this higher level processing within the insect brain. Right, so now we're going to look at some of that real data. So to prep you what you'll see on the next slide, so this will be a slice, or like a very thin slice of neural tissue. So you'll see a lot of cell bodies. Highlighted in orange will be a single neuron, and we'll kind of watch a video of how this single neuron's kind of traced in 3D space. All right, so this image is taken via an electron microscopy. And so this is a technique that allows incredibly high resolution of tissue, cells, and you can actually see cell bodies or organelles within these images. And so what you're looking at is a single plane within the odor processing region of the Drosophila melanogaster brain. And so in orange is a single olfactory receptor neuron, and you can see that, and what we'll do is we'll trace it when we play this video right here as it's moving through different planes, right? And so that's on the right, you can see that as we're moving through this plane, we're actually creating that volumetric structure. The way that this kind of data set was created is taking, again, those thin slices of neural tissue, computationally stitching images together that have overlapping regions. And then as you're moving through different tissue preps, right, you can follow the outline of that single neuron through like that third dimension, right, giving it that volumetric structure. Yeah, this is an incredibly cool technique. This was led out of a group in Genelia. There's collaborations with Google, University of Cambridge, and a bunch of other big academic institutions. Yeah, and this took, over a decade, well over a decade to get this data set, and they have the complete connectome of the Drosophila melanogaster brain. It's the largest organism to have a complete connectome, which is mapping every neuron within the fruit fly brain to this level of resolution, which is pretty incredible. Yeah, so this is how we can kind of create these volumetric structures, and it doesn't need to be this high resolution. Again, to orient us, So we're looking at the Drosophila brain, and that fits right here within our Drosophila. We're going to again look at those information processing centers and just touch on four that I think are crucial and conserved across different species. All right, so we'll start at the top going down. We'll start with the antennal lobe. And this is the first region of odor coding within the insect brain. And so you can orient yourself by thinking of the antennae, where those olfactory receptor neurons are sitting, and they project to this first region of odor coding within the insect brain called the antennal lobe, right? And that's where that sensory information is turned into an odor identity, right? Um, Next is the mushroom body, so that's the center of learning and memory in insects. And so this was the first image I'd shown of the mushroom body in Drosophila, right? And this can add kind of positive or negative valence, that is like thinking of approaching or avoiding a particular stimulus. The mushroom body is often very strongly associated with the antennal lobe and olfactory information. But there is some input from visual systems as well. Which leads us next into the optic lobes, right? So these are like first region of visual information processing, right? And their main goal is to take information from the structures that make up the compound eye in an insect called ommatidia that have a bunch of different photoreceptors that are responsive to light, right? And so that they turn that stimulus into usable information for your insect to create a composite image, but also helps with motion detection, classifying shapes, colors, again, for whatever the goal of your insect. The last structure is called the central complex, and this is a really, really interesting structure in the brain that's extremely well conserved. Yeah, it's important for integrating other sensory information and creating odor output. People call it kind of the brain of the brain. It's critical for movement, orientation, path integration. When we think of those monarchs that are migrating across multiple generations, you can think of something kind of innate in the central complex that allows for the orientation of the migration and orientation of different visual stimuli for such long periods of time, and that being encoded innately in the central complex. All right, so we're going to end on just kind of taking those four structures we just talked about and comparing across four different species. So here, again, the four colors correspond, roughly four colors correspond to the four different regions we talked about. So blue is the antennal lobe, green you kind of see tucked in the back is the central complex, yellow on the outside is the optic lobes, and then the mushroom bodies are in the orange structures that you can really see in the bottom two, apis and bombus. Yeah, and so I hope to illustrate the idea that these structures are conserved across species, but there are significant differences, and think a little bit about how that affects behavior, maybe how like form fits function and function can fit form, vice versa. So quickly listing the species, starting at the top left, Manduca sexta, that is a hawk moth. On the top right, that is a mosquito. Bottom left is Apis mellifera, so honeybee. And bottom right is Bombus terrestrius, bumblebee.